Well, welcome back to Unit 4. Unit 3 is done and dusted, and away we go. So, this first part's all about, one's all about how species related, and this first unit within that is about the changes in the genetic makeup of a population. And that's not responding. Why is that not responding? Come on, that's better. So, the contents of this particular section is quite intense. Lots of very interesting stuff. So, essentially it's all about allele frequencies. So allele frequencies are the most important factor in determining change, and we're really looking at, well, how do you change those frequencies? So we look at mutations and abnormalities to chromosomes. There's all about natural selection, the way species arise, this idea of genetic flow and genetic drift, which is really allele flow and allele drift. We then look at the importance of biodiversity. And obviously, selective breeding comes into play because it tends to impact about how well we can, how we can influence allele frequencies and maybe make problems rather than benefit things. So it might take two videos. We'll see how we go. So most importantly, what is this idea of genetic variation? Genetic variation essentially is all about how many alleles are present for any particular gene. And the more there is, the better, basically. So we pass this on via sexual reproduction, which is why sexual reproduction is preferred to asexual, because it's mixing of genes. And new alleles arrive by mutations. So you are, in fact, all mutants. And I say that lovingly. We are all mutants. We're all variations. I can look at you as a class of 20 young people, and you all look different. And those differences are an accumulation of mutations over a long period of time. So, lots of terms we need to make sure we're aware of, and now those of you who have done Units 1 and 2, this shouldn't be anything particularly new. In fact, you should know some of this from Year 9 and 10, but just to, again, go over some stuff that you should know about. So genes, of course, are those functional bits of DNA, and they code for a protein. The allele is the form of a gene. So, of course, you have two copies, one from each parent. But any particular gene can have many more than two alleles. But you only have two yourself. And each allele will produce a functioning protein. Now, some proteins will blend, and we get that codominance happening. And in other cases, one will dominate the other and stop you seeing it in the phenotype. So genotype is describing the alleles present at any particular locus. And I've got the example there of big A, little a. So a heterozygous individual, and the phenotype is the action, as it's the proteins that have been expressed, and that could be blue eyes or red hair or whatever. Then we talk about frequency. So we only have two alleles each usually. We'll talk about when we might have more than two, a bit later on. And we inherit one from each parent. So we actually measure how common each allele is in a population by measuring the frequency of that allele in its availability, and often it's done via phenotype, so it's assumed rather than directly measured. But of course, with genetic testing, we now directly measure that as well. And sometimes we end up with what's called a fixed allele. So for whatever reason, for a particular locus, a particular characteristic has been really beneficial, and those who carry maybe the recessive trait disappear completely, and you're left with just one allele, and there's only one look within a population. It's not truly fixed, but if we think of achondroplasia, for those who remember their Year 9 studies, is a condition of the heterozygote. So you are big D, little d. I can tell you that everyone in the classroom is homozygous recessive. Homozygous dominant is a lethal combination, that person dies, and the heterozygote is a dwarf, which would suggest that dwarfism was probably more frequent and regular when we were living in caves and climbing in trees and things, and it probably had great advantage then. But we've moved away from that, and we've almost fixed that gene so it doesn't exist in that population. So it's a minority sort of condition. I'll keep moving because I've only done 2 slides and it's already 4 minutes in. So biodiversity, well, we talk about biodiversity a lot, don't we? And biodiversity is one of those really, really important things. We like to have lots of animals and plants present in an ecosystem, because that suggests that ecosystem or that habitat or that environment is healthy and has the ability to adapt to change more successfully. But that biodiversity is really importantly determined by genetic diversity, so that you want lots of species, but within each species lots of genetic variety. So there's variability within each population of each species within your environment. And then we're really talking about allele frequency. So what we're talking about is high diversity of alleles. So for each of the loci of each gene, you want, many alleles present. And that gives you a much stronger, healthier population, because you have the ability to adapt to change. Extinction generally occurs when a species can't adapt to change. So most of that major, and there's been 6 major extinction events on the planet, has happened due to some massive climatic change, and species haven't adapted fast enough. But normally species will adapt and change with the environment, and maybe a new species arrives and the original species disappears, but that ability exists because of the diversity of the alleles. So biodiversity is really about allele diversity. The chap there with that fantastic beard is a guy called Charles Darwin, who you may have heard of. And we're really talking about Darwinian evolution. It's been refined and developed since his time, as you can imagine, but essentially what Darwin was on about, and what we still talk about, is that for a species to evolve, there needs to be variation with respect to various traits. So you need lots of different alleles. Some alleles will be better suited to survive in the environment. Not all individuals get to survive and reproduce. Those that do will be those with the alleles that confer success. So they're the fit ones for the environment. And over time, these traits will slowly increase, and we'll see big fluffy beards becoming dominant or something. That still holds. They're basically Darwin's original thoughts. He didn't know about genes or alleles, but he was right. He didn't need them in the end. So a little quick reminder, because there's some things we're going to need to talk about from Unit 3 and even earlier in the course that come into play here. This Unit 4 really does use a lot of the stuff you've already studied in application. So remember protein synthesis. Hopefully you remember protein synthesis. Oh, teeth back in, because this is going to be on your exam. So DNA nucleus is unwound, and of course helicase is responsible for that, and the template strand is transcribed onto messenger RNA, remembering to remove the introns and keep the exons. They will become important a bit later. DNA ligase, then of course will close it up. The messenger RNA will leave the nucleus via the nuclear pores and travel to a ribosome, where messenger RNA is translated by transfer RNA into a polypeptide chain of amino acids. And we see it happening nicely in the diagram there. It will then coil, pleat, fold, and become a protein with an active site in the endoplasmic reticulum. We read DNA as triplets, 3 nucleotides at a time. We read messenger RNA as codons, 3 nucleotides at a time, and transfer RNA as an anticodon, 3 nucleotides at a time. So stressing that because they're going to become important. But then what happens if something goes wrong in transcription and translation? And sometimes it does. And we get mutations. We tend to think of mutations in the popular press as something really nasty and horrible. But of course, we are all mutants. And without mutations, we don't get variation. Some work, and some are really useful, and some are of no value at all. So there's some lovely flies here. With poor old fruit flies get stayed a fair bit. They breed fast. And you can see the top corner there. I'll get my little pointer. Here's a normal fly. Fly quite happily. This one has enlarged wings, so probably not disadvantaged. It's probably fly faster maybe. Well, they've got to flap them even harder. Maybe it takes more energy to flap them. This guy's got little tiny wings. That may be much use. This one seems to have a different wing shape again. These guys' wings aren't in great condition. And then, oops, no, I'll get me out of the way, didn't I? Anyway, I'm in the way there, but this one hasn't got wings at all, so no value at all. But there's 6 wing types, 1 gene loci, and some of them are useful and some of them aren't. So we talk about point mutations. And there's some point mutations in humans here, the hip dysplasia and knee joints and toes. So it can be a silent mutation. There's point mutations. A mutation is a change at one nucleotide position. And they can be silent, so it doesn't change the amino acid produced. So it doesn't change the protein. And you have heaps of those in your body. It can be missense. So it caused a change to the amino acid that has changed the protein, but that might mean you've got these big gaps between your toes. Yeah, is that going to cause any pain? Not really. Strange looks on the beach, but otherwise okay. Or it produces nonsense. The protein produced is not functional at all, and they're not much value to us at all. So these point mutations can be a substitution. So here we see this A has become a G. So tyrosine is now his. Can't remember. So we changed one amino acid. This may have absolutely catastrophic effects, or next to nothing. This could be a blonde person now giving rise to a child with red hair. You know, it could be a really minor thing. It just could be a new variation, which is fine. And I suppose most variations that we've developed are going to be these sort of substitution mutations, which have just caused a small change. We can also see where a base is inserted. So we've got ACAT there, AGCA. So by putting a G in there, we've changed valine to arginine, but downstream, all the way down here, we've now shifted. It's called a frame shift. We've now shifted every reading triplet. So now creating a whole range of different amino acids. Now again, this could work out to be okay. We could get a nice, useful new protein. More likely it's going to not work. It's going to be a mess. That's one way of, that's a point mutation, it's a single nucleotide. And again, same with the deletion. So in this case, I've taken the C out. So ACA became AAT. I just threw an A in there to put something in. And again, my valine has become tyrosine, and everything downstream has been shifted. That frame shift has occurred again, and all the amino acids are now wrong. And again, we could have a catastrophic problem, or we could have something new that's more likely to be catastrophic because you're going to cause so many changes. Particularly as I've started with the start instruction here. We're at the third amino acid in a protein. So the frame shift is going to be thousands of amino acids long and cause all sorts of problems. And you also have block mutations, where great chunks of the chromosome get moved around. And we can do a number of things. We can duplicate. I pinched this from Ed Rollo, so apologies to Mark. I pinched your graphic, sorry buddy. So a block of that chromosome has been copied again. So we've got second copy. Yeah, second copy of the gene, may be problematic. This one here is an inversion. May have little impact at all. You just change the order of them. Here we've seen a deletion of a chunk of chromosome, and so the chromosome is now shorter, so we have lost a complete gene or many genes. Here we've seen an insertion. So this is what we said, that chunk there has been inserted into this particular chromosome. That chromosome is now longer, this chromosome is shorter. Not a major problem, really, because the gene's still there, unless you're making new gametes, and then you've got new variations. But in your somatic cells, this is not a big deal. And same with translocation. I've taken a chunk of this DNA and I've moved it over there. So what? In my somatic cells, the genes are still there. If it's chromosomes, well, we're starting to make new information, which is particularly useful. I might stop here and start again. We've 14 minutes, so I'm up to the second half in a second podcast. Back in a tick.